Hello and welcome to this Integrated DNA Technologies webinar on planning and executing siRNA experiments, good practices for optimal results. My name is Hans Packer and I will be serving as moderator for today's presentation. The presentation will be given by Dr. Garrett Rettig, who is a research scientist in molecular genetics at IDT. Garrett has a PhD in pharmacy and medicinal chemistry from the University of Iowa, where he studied synthetic peptide delivery systems for plasmid DNA and siRNA, both in vivo and in vitro. At IDT, Garrett has been involved in high-throughput screening of siRNAs in vitro and has recently co-authored a comprehensive review of siRNA in vivo and published this in Molecular Therapy. Garrett's presentation should last about 25 minutes, and we will follow that presentation with Garrett answering any questions you have. As attendees, you have been muted, but we encourage you to ask questions or to make comments at any time during or after the presentation by typing them into the questions box, which you should find located at the right-hand side of your screen. At the end of the presentation, I will direct your questions directly to Garrett. Also, in case you need to leave early or want to revisit this webinar, we are recording the presentation and we'll post it on our website or on our YouTube channel at www.youtube.com/IDTDNABio. Now, let me turn over this webinar to Garrett for his presentation entitled Planning and Executing siRNA Experiments, Good Practices for Optimal Results.
Thank you, Hans. Good morning and welcome everyone to the webinar this morning. Just by way of further introduction, I want to mention that the webinar was set up and the information included this morning for someone who maybe has some familiarity with siRNA or the use of RNAi in vitro, but would like to, has a specific application in mind perhaps, or would like to use it a little more extensively. So we'll be addressing some key points from that standpoint. Additionally, I'd like to highlight some of the bullet points here as a further introduction. So we'll be talking about such things as understanding the target transcript along with siRNA selection, which, as I'll talk about a little bit more, are a little bit more of the trivial points to this whole process. And then when we get into the actual experimental, we'll be discussing choosing which cell type might be best, validating the assay, which of course is important, and then including the appropriate biological controls. At the bottom of this first slide, you can see that I've included schematics of the dicer substrate siRNA or the dsRNA, also the canonical siRNA. So the dicer substrate you can see is a 25-mer sense strand and a 27-mer antisense strand. And of course, I'm sure you're familiar with the canonical 21-mer with 3' overhangs of the siRNA. By and large, I'll be using them interchangeably throughout the discussion, both because they are synthetic RNAi triggers. And then just to narrow the scope a little bit more is that we will be discussing everything from the standpoint of using synthetic siRNA in vitro experiments. So this first slide you would probably recognize as an amplification plot. This happens to be a qPCR assay designed against a particular gene of interest, a hypothetical gene of interest, which we'll be discussing throughout. In that particular gene of interest expression in HeLa cells. If we were simply looking at this data following an experiment, we would see that the untreated controls compared to the amplification from siRNA-treated cells, we see about a 3.3 shift in the CQ, which we would predict, which would calculate out to be a 10-fold decrease in expression based on siRNA treatment, which would correlate to a 90% knockdown. The question is, looking at this data, is that enough in itself to convince us that the knockdown is real? Might there be some toxicity associated that's leading to this? Is this a false negative in some other way? Conversely, it could be true that we would see no knockdown, but we could have a false positive situation. So going forward, we'll address some of the controls that we can include and some of the considerations to keep in mind as you go through in setting up an experiment that would allow you in the end to confidently say whether or not this 90% knockdown that we observe by this qPCR amplification is in fact real. So first, with talking about the transcript that we're targeting. The data below is from a screen, a high-throughput screen that we've done in-house. So you can see it's about a 5 kb gene of interest that we're looking at expression and knockdown in HeLa cells. In this particular experiment, we generated about 300, greater than 300 siRNAs against this target that are all spaced just randomly at about a 12 to 15 base frequency. One thing to point out are all the controls involved, and we will get into these more in detail. But in the title, you can see that these are in HeLa cells at 1 nanomolar. We've included normalization to 2 human reference genes, human HBRT and SFRS9 as well. We've included setting these knockdowns to the 100% level of a negative control 1, 5, and 7 that we commonly use in our experiments. So this is just to highlight some of the controls that will come into play that, as I said, we'll discuss a little bit more later. But in terms of the secondary structure and protein binding considerations, along the x-axis here, we sort of see a schematic of what the transcript looks like. However, of course, we know that it's not a nice one-dimensional line that is lying around inside the cell waiting for a RISC loaded with an antisense targeting some particular sequence in here to come in, cleave, and knock down the messenger RNA. Rather, there are regions of secondary structure, also protein binding domains that might inhibit RISC activity. And so you might think that some of the areas where we don't see potent knockdown could certainly be areas of secondary structure. By and large, one other thing to consider is that this collection of randomly generated siRNAs does a pretty good job of providing some very potent sequences. However, going forward, we'll talk about some ways that you might, better than sort of a random scatter shot, better ways to select a potent siRNA. In terms of the transcript, one other thing to consider, and just an aside here, all of these things that we'll be discussing about today in the webinar are based on experiences either we've had in our high-throughput screening or experiences that our collaborators have had as well. So some of these things may seem trivial, and in fact they probably should be if you're thinking completely about an experiment, but we've seen them happen, they can happen, and so I think they're very much worth mentioning. So in this particular case, we're showing here a hypothetical targeting of mouse GAPDH. And this information of the mouse GAPDH transcripts was pulled from MGI. You see the website down here, this particular database of genetic information from mice. What we've seen happen in the past is that depending on the cell line that's used, the mouse cell line that might be used, there might be different transcripts expressed at different frequencies. So for instance, the second transcript here, which has a unique exon, is not as prevalent as the first transcript, but you design an siRNA or choose an siRNA that targets this unique exon, yet your qPCR assay is looking for and amplifying in a region that is unique or that is common amongst all the transcripts. You might be able to generate a very potent, and you might in fact be using a very potent siRNA, but it's knocking down a less common transcript, and so your qPCR assay is going to pick up the unknocked down and the uninhibited transcript, which would certainly lead to a false negative result here. One other thing to consider regarding the transcript is that, and again, this may seem quite trivial, but we've seen it happen, where perhaps the experiment in your lab is going forward and you want to generate and validate in vitro a potent siRNA against a particular gene of interest. And your lab has had some familiarity with using HeLa cells, And so if you test siRNAs against in HeLa cells and have a validated assay and are confident that you've got a potent siRNA, but it so happens that the siRNA happens to be in a non-homologous region between human and mouse. So then perhaps the experiment goes forward. You want to look in vivo. Maybe you have a vivarium accessible to your laboratory, or you work with a collaborator, hand off the potent siRNA that you've validated in vitro, and all of a sudden you've got no knockdown being observed in your assays from the mice that you're dosing. So suddenly you've gone to in vivo assays and you start chasing various other variables that you think might have gone wrong, whereas really it's just this simple question of species to species variation. So just in general, certainly understanding the transcript is an important point to consider at the outset. Then in terms of selecting an effective siRNA, you can see these manuscripts that I've noted here, all published in the early 2000s, that provide some design rules based on empirical data. These groups went through and identified various rules of GC content and not more than 4 bases and certain sequence motifs that would be most desirable for generating or selecting a potent siRNA. And all of these are, I think, very good methods and mechanisms to use. However, today we find that you can go online and find various web-based applications that will aid in selecting a potent siRNA. Some are based on the design rules shown here. Others are based on algorithms such as automated neural networks and support vector machines. The algorithm that is in use here at IDT is based on an SVM model. Briefly, the way that we use this is that given an accession number or a gene symbol, this pre-designed algorithm will generate what the algorithm dictates as the top 10 most potent siRNA sequences. Of course, these would be dicer substrate siRNAs. The top 10 most potent predictive sequences, and we would send 3 of them to the end user. The guarantee would be that 2 of the top 3 dicer substrates would exhibit, as you can see here in bold, greater than 70% knockdown at a 10 nanomolar transfection in a well-controlled experiment. In order to look empirically at this guarantee, over the past year or so, we've tested in-house over 50 genes to try and confirm what this frequency is. And in our hands, 42 out of 50 genes, or pretty high, 84%, had 2 of the first 3 that did knock down the target at a 10 nanomolar transfection. And in fact, then, if you looked at all the top 10 for each of the 50 targets, 50 out of 50 had at least 3 dicer substrate siRNAs in the top 10 that achieved greater than 70% knockdown. So moving from selecting an siRNA to selecting a cell type, now we're training more towards actually getting to some experimental work here. This first example, this first slide for selecting a cell type comes from BioGPS, which is a fairly useful database that we've incorporated, I think, quite a bit in our own research in terms of trying to identify at the outset of an experiment which particular cell type or which tissue type in general might have relatively high abundance of our gene of interest. This particular plot happens to be for human GAPDH and shows the tissue prevalence with, again, the various tissue types and cell lines along the x-axis. This has saved us some work in the past by doing this sort of computer-based pre-screen of cell lines. It has directed us to look more specifically at particular cell lines that we might have in stock in our own liquid nitrogen stores, and also it has directed us a few times pretty successfully as to which cell lines might be good to purchase from, for instance, ATCC, or to obtain from a core facility that would have predicted relatively high expression of our gene of interest, our target of interest. That's not to say that there really couldn't be any substitute for literature search. In this particular example, I just did a simple PubMed search of siRNA GAPDH in vitro and came up with this recent manuscript from Biomaterials in which the authors are testing a novel delivery device. So they won't discuss which particular delivery reagent they're using because they're using their own test material. But we can quickly read in the materials and methods that they are targeting GAPDH in mouse fibroblasts. And we see the seeding density, we get an idea of their dose response that they're using for siRNA delivery, and also that they assay by qPCR. So again, this is just I think we all need sometimes reconfirmation or affirmation that literature search is oftentimes a very valuable use of time. And then regarding an assay selection, there are several assays that could be used to look at siRNA-mediated knockdown. The one we prefer, as you've probably already noticed, is qPCR because of the low limit of detection, high sensitivity, and large linear range of detection, over 6 orders of magnitude. You could also use Western blot or a bDNA assay. Western blot, of course, would look at protein, or northern blots, or even look at the phenotype. One thing to note here is that in validating an assay, you want to see that your selected cell line and untreated controls in that cell line are giving you a reasonable signal that's well within your linear range of detection, which we see we have here. And you also want to consider that upon treating with your siRNA that you're going to see up to 90%, 95% knockdown. So you want to be sure that given a tenfold decrease in your signal, you'll still have a reasonable signal-to-noise ratio and can be confident in the data. And qPCR provides us with that. One item to note in terms of assay selection, this data comes from a Nature Methods paper in 2006. We're looking at HPRT, both mRNA knockdown in the left panel and protein knockdown in the right panel. The mRNA knockdown was assayed by qPCR, and what you can see over this time course is that after 6 hours there's about 10% remaining, and after 12 hours it's below 5% of the of the transcript of the messenger RNA remaining. Of course, protein follows the transcript and phenotype follows protein. So when we start to consider the time course at which perhaps to look at Western analysis, at 24 hours, we're still only seeing about 60% knockdown. 48 hours, we're down to about 20, and then finally out to 72 hours, we see sort of the same results that we're seeing already at 12 hours when we're looking at the messenger RNA. So this is just to show you that it's important to consider the time course when deciding on an assay in which particular biomolecule that you are assaying to determine your knockdown. And now that we've gone through and hopefully identified a potent siRNA, selected a cell type, it's the question of how are we best able to get that siRNA into the selected cell type in order to mediate the knockdown that we're looking for. There are several products out there available commercially for use. Most are lipid-based, some are peptide-based, or even some polymers that can be used to effectively deliver siRNA. One example, again, that I'll show here is that oftentimes we'll go through and we'll screen various, you see over here in the legend, various reagents against a new cell type, and U87 happens to be a glioblastoma cell type. We're looking in this particular experiment just at HPRT, which is our transfection positive control, HPRT knockdown, normalized to SFRS9 in a 24-hour reverse transfection, something that's that we routinely do, and we're varying the transfection reagent in use here, and we can see that all of these particular reagents would advertise that they can, in a broad spectrum of cell lines, knock down a target of interest. We see that there can be quite a discrepancy. We see about 65% knockdown for interferon in this particular cell line, which sometimes may not be bad, but when it's compared to, for instance, the TKO or the silent effect or the RNAi-max, those are quite potent, and we're seeing greater than 90% knockdown in those cases. So this is just to illustrate that it does bear the time to screen a few different reagents if you have that capacity to find which one is optimal in delivering siRNA. And then experimental variables to consider when optimizing this particular experiment. First, in the dark blue at the top, I've sort of highlighted the parameters which we find in our high-throughput screening to be to have the most impact in optimizing the transfection. So, for instance, if we're looking at different cell lines, we always screen a variety of reagents, as we just showed, because those can have quite a variable effect. Also, there would be a titration of a reagent dose response to see sometimes if there's too much reagent, you can see some toxicity. If there's too little reagent, the siRNA doesn't complex well and might not deliver as effectively. And then cell seeding density is something that can be based on previous experience with a similar cell line, a similarly sized cell line, but of course this is important in terms of avoiding some toxicity issues in some cases. Also, it's important in terms of being able to downstream have enough signal in your endpoint assay of your gene of interest. Other variables that are in play and may certainly bear optimizing would be an siRNA dose response. In our case, typically we're looking at 100 picomolar doses up to 10 nanomolar doses, so that's pretty fixed in our general workflow of the screens that we do in-house. The transfection time course could also be variable, as we discussed, depending on your assay, although typically we are looking at a 24-hour transfection, and a 24-hour transfection in a reverse is our preferred mode of operation. A reverse transfection being that the siRNA complex with the reagent is added to the cell suspension, and then that transfection is carried forward 24 hours from that point, as opposed to a forward transfection where the cells would be plated for 24 hours in advance of adding the siRNA complex with the reagent, which would then be allowed to incubate for another 24 hours, so it would be a total of a 48-hour transfection in effect. And then also another thing to consider, especially if you're working with a peptide-based system or a polymer-based system, would be what is the reagent to siRNA ratio, or what is the so-called N to P ratio that may in fact have a dramatic effect on the results. And as we're going through optimizing these particular variables in setting up the transfection and moving towards actual experiment, we will typically include, as I mentioned on an earlier slide, 3 negative control isosubstrate siRNAs that we've validated in several cell lines and across several targets not to have any off-targeting effects that we've observed. And also, we commonly use positive controlled isosubstrate siRNA HPRT, or the hypoxanthine guanine phosphoribosyl transferase targeting siRNA. This particular isosubstrate targets mouse, human, rat, and Chinese hamster HPRT. And we use all of these factors then to come up with our optimized conditions for experimentation. So this slide would be an example of the experimental setup where we would get to the point of actually testing a gene of interest. It might look a little silly to include all these controls, testing only one gene of interest, but in this particular case, we've shown in our experience that we can't be confident in the results unless we've validated that cells only negative control, reagent only negative control, and also negative control siRNAs transfected in at 10 nanomolars produce no observed knockdown in any of the reference gene assays or the gene of interest assays that we'll look at by qPCR. Likewise, we want to include certainly a positive control, and as I mentioned, we use HPRT as our positive control dicer substrate that will transfect in a dose response in order to validate that our transfection is quote-unquote good. Typically, we use HeLa cells in our high-throughput screening, and this 10 nanomolar result would be about 5% of the HPRT remaining after 24 hours, and maybe 10% for the 1 nanomolar and about 20% for the 100 picomolar dose. But that gives us a nice reference of, was our transfection efficient, and can we rely on the data that's coming from the gene of interest that's showing that the knockdown that we're observing. One other important point is that this, of course, in a 24-hour well plate is showing 8 different treatments, all with biological triplicates. Going forward from this point, in our particular case, we would be isolating RNA, going through then reverse transcription in qPCR. So there would be technical replicates along the way as well. In fact, most notably along the lines of replicates on the qPCR, in order to ensure that not only are our biological replicates consistent, but also our technical replicates consistent in the final data output. So then we get to the point where we're sort of coming back to the first slide that we looked at in the beginning, but first we have the advantage now of looking at, in this particular amplification plot, HPRT expression and the dose response that we talked about. So we see the negative controls in the gene of interest siRNA, which we expect not to affect this particular reference gene. And we see a nice knockdown at 10 nanomolar HPRT transfection, slightly less at 1 nanomolar, and then at 100 picomolar, a nice walk down in the dose response. So at this point, we're confident that the transfection itself was efficient. It's also important to look at our other reference gene, which is the human SFRS9. Those expression levels are consistent no matter what the treatment, negative control, gene of interest, dicer substrate, or HPRT at whichever dose. So this is a very much expected result for SFRS9, a nice stable level for us to normalize to. And then we will go forward and look at our gene of interest assays. And I say assays plural. We typically will look at 2 different assays for our gene of interest in a given screen. One would be at more the 5' end, the other more at the 3' end, to give better geographic coverage of the entire transcript. So we see nice knockdown of our gene of interest treated cells at 10 nanomolar transfection compared to the negative controls and the HPRT treated cells. Likewise, the second assay would give the same result. So at this point, we've circled back to the slide that we started at, but now can say with much more confidence that the knockdown is in fact real, that we are observing in our experiment here 90% knockdown of our target. And one final slide to really highlight what I was mentioning about the geographical coverage of a transcript with multiple qPCR assays. You can see that the first assay, more at the 5' end, is represented by the red data points. The second assay, more at the 3' end, is represented by the blue data points. At the 5' end, there's much assay concordance. The red and blue dots are typically on top of one another, but as we move more 3', we see quite an assay divergence. So if we were only, looking at the 5' assay, we would observe almost no knockdown for siRNAs that are near the 3' end of the target, when in actuality, when using our 3' qPCR assay, we see that these, in fact, are quite effective siRNAs and give some of the most potent knockdown that we see across the board here. So this is just sort of a technical aside, but something to consider whether you're using qPCR assay or even other assays, that you might want to have more than one level of assay validation. So just finally, in summary, this is a flow chart that sort of takes you through from the standpoint of first identifying your target of interest, considering variations in the transcript, whether it be transcriptional variants or species to species variation, regions of homology, etc., as we discussed. In concordance with that is really the siRNA selection, both which feed into selecting a cell line. Cell line selection really needs to be done in parallel with assay validation before you can move into having a controlled pilot transfection, and then you would move through optimizing the conditions that we discussed in order to have finally conditions that you could be confident are optimized in order to look at your gene of interest and the knockdown that you might be desiring. So with that, I list a couple of resources here of IDT's online tools that can be used to generate dicer substrate siRNAs. Also, these are publications from our group, from our research group here at IDT, that especially the Nature Methods paper, which would give some advanced and more detailed background of setting up and optimizing transfection conditions. So with that, we'll take any questions that you might have.
Okay, Garrett, thank you for that great presentation. And at this time, we do have some time for questions. So if the audience has any questions that they would like to ask, they can type it into the questions box, which they should see on the right-hand side of their screen in the GoToWebinar software. And there are a few questions here for you. So let's get started. Garrett, what is a reasonable range of concentrations for someone to try for a dose-response curve in a cell type?
Typically, what we see and what we've noticed in our experience is that using lipid transfection reagents, we are typically looking at 100 picomolar up to 10 nanomolar concentration of siRNA. That's for a lipid reagent. We've also tested different peptide reagents that have required us to go up to 150 and 300 nanomolar range. So it really does depend on the reagents you're using. It also can depend on sort of, what amount of reagents you're using, as well as the ratio of reagent to siRNA and how that might affect toxicity in the cells. But the most general answer I can give is that we are typically using 100 picomolar to 10 nanomolar, and if you're not using a lipid reagent, then you may, which in our hands tend to be the most effective in vitro, then you may have to go to a higher concentration.
Okay, the next question is, this person is new to doing siRNA experiments, and they want to know how long it takes for somebody to get from that initial stage of designing a set of experiments to, all the way through to actually carrying out and collecting their data for a paper.
Yeah, so if you've got maybe a hole to fill in a publication, and of course you would have the target identified, I think, and all these transcript considerations and siRNA selection can be done sort of without too much bench work. But then it comes to assay validation and cell line selection, which can take up to a couple weeks if you're pretty efficient, I think, at finding a cell line that works well for you and then validating an assay that you're confident in. And then I would say maybe another week or two, depending on your level of experience with tissue culture and how efficiently you move through the experimental process to hit on the optimized parameters, and also depending on how long it might take you and how many different variables you might have to look at in order to come up with some sort of knockdown that you perceive as good as in your endpoint assay. So I would say probably best case scenario would be about a month of bench work to go from having a target in mind and selecting an siRNA to having an optimized experiment where you can produce reliable results.
Okay, we have another question here. It says, could you compare the knockdown success rate between IDT's siRNAs and chemically changed siRNA oligos like stealth technology? Not familiar with that second term.
Right. So one of the things about that comparison is that the stealth technology, for instance, as the listener points out, has various chemical modifications. We can also build in different chemical modifications into our dicer substrates as well that would enable those sort of non-immunogenic responses and inhibiting of off-targeting effects to occur as well. But we've seen very much on par results in terms of potency comparing our dicer substrates to stealth siRNAs, and in fact, the most basic comparison is an unmodified dicer substrate siRNA compared to a canonical unmodified siRNA, and we've consistently seen improved potency, especially at doses at 100 picomolar and lower concentrations. We've seen more potent and more efficacious results from the dicer substrates in our hands.
This may be a question that could kind of be a good follow-up to that, which is it's about the time course of an si experiment. So if the time course of an siRNA experiment, if the effect is too short, is there anything I can do to make the response more stable or long-lasting?
One thing that you could do if the effect was in a more narrow time window, I mean, one of the slides that we showed previously did look at a time course, and so one of the things that you might want to optimize is where's your knockdown most potent, and if it's at 12 hours, you could stop the transfection at 12 hours, isolate the RNA at that point, and then go forward and have your most efficacious time point at a time that was earlier than 24 hours, I think.
Okay, and here's a question. You've shown some data in your screens that involved hundreds of siRNAs. How long does it take to carry out one of those types of experiments?
Yeah, so we've been doing these types of high-throughput experiments for the last 4 years and have really, over that time, I think we've screened 75 different targets and have cumulatively thrown over 10,000 dicer substrate siRNAs at these various targets. So I say that to sort of say that we've had a lot of experience with this, so it's not something that someone could pick up and do at the outset. In addition, we have liquid handling robotics in play that we use to isolate the RNA, to set up the reverse transcriptions in 96-well plates. We do the transfections in 96-well plates, everything downstream to RTs in 96-well plates, and then qPCRs in 96-well plates, and then have robotics on thermocyclers in order to load those instruments. So we've got the infrastructure in place to do all that, and it works quite seamlessly for us. It would typically take us about a month to, once from the time that we got, that we identified a gene of interest, to generate and synthesize the dicer substrates here at IDT, and then to actually carry out the experiments in the lab and generate data would be in the course of about 1 month. But again, we've been at this for a little while, so we've got our methodology pretty fine-tuned, I think.
Okay, here's just a quick question about terminology. You mentioned in one of your slides something called a reverse transfection, and I believe that was with optimizing for your
right.
transfection reagent. And I just want to know what the term reverse transfection meant.
Yeah, reverse transfection is something that we do that primarily in our high-throughput screening because it cuts 24 hours off the transfection time. So in a typical transfection, which we would call a forward transfection, cells would be plated in a 6-well plate or a 96-well plate, what have you. For 24 hours, cells would become adherent and grow more confluent. Then after 24 hours, you would complex your siRNA with your lipid reagent of choice and then add that to the 96-well plate and then carry out the transfection from that point, an additional 24 hours, which gives you a total of a 48-hour time window. Whereas in a reverse transfection, we found it to be just as effective in terms of knockdown, potency of knockdown, to add the siRNA complexes, meaning complex with the lipid reagent, to the cell suspension in, for instance, the 96-well plate. We add those both at the same time. Cells become adherent, also take up the siRNA complexes at the same time, and we're talking then only 24-hour time window from beginning the experiment to harvesting cells and isolating RNA or whatever the downstream application would be. So we've really found a time advantage of using reverse transfections and have not seen any loss in terms of potency of the transfections.
Okay, there's one more question here, which is, and keeping in mind that this webinar was presented for in vitro siRNAs, how is the workflow different for in vivo experiments?
Well, I think it would be quite a bit different. One thing that I can say is that in a lot of our in vitro screens, in some cases anyway, we are building towards in vivo experiments. So we will build in different chemical modification patterns in order to, going forward, dampen an immune response or provide more stability to the siRNA. So these are factors that might want to be included in the in vitro experiments with an eye towards specifically moving towards in vivo siRNA experiments.
Okay, and that seems to be all of the questions that we have at this time. So I want to thank Garrett for your presentation today. And this is one of a series of webinars that IDT will be presenting on siRNA as well as other topics in the near future. And we'll be emailing you about these future webinars as they're scheduled. All of the previous recorded IDT webinars can be viewed at our YouTube channel at www.youtube.com/IDT-DNA-bio, and they're posted on our website under the Support tab. And thank you again for your participation, and best of success in your research. Thank you very much.